student, I'm Mr. Lam. So in this video, we are going to talk about the reading to learn again. And remember what I mentioned before, we do not only memorize all what we learn, we also need to apply what we learn in our daily life for any biological phenomena around us. So this video, we are going to talk about the biotechnology for reproduction or growth in development. So remember, we have already learned chapter 12, 13 and 14, of course also 11. Okay, so when you do the revision, remember to do the revision for chapter 11, 12, 14, 13, 14 together, because they are just the 4 episodes in one movie. So the first part we talk about the cell division, and the second part we talk about the sexual and asexual reproduction, the plant reproduction, and then the human reproduction, and the last part will be how can an organism grow and develop. So in this video, because we have already talked about the chapter 11 before, so in this video we only talk about the chapter 12, 13 and 14. So for the biotechnology part, how can we use the biotechnology to reproduce the plant? How can we use the biotechnology to help some couples to undergo the in vitro fertilization to have a baby? And how can we use the biotechnology to save or to store the seed for to maintain the biodiversity in the ecosystem? So the first part, we talk about the tissue culture first. Propagating plants by tissue culture. Tissue culture is a type of biotechnology, not only for the crops or the horticultural plants. They can be propagated vegetatively by method called tissue culture. So in this part, I want you to recall that vegetative propagation is a kind of sexual or asexual reproduction, and in this method, it's only a small piece of tissue from a parent plant is grown. Okay, that means we only cut a small piece of tissues is okay. But the point is that just for the small piece of tissue, we need to know which tissue we choose. For example, okay, will you choose the xylem vessels for the tissue culture? No, absolutely not, because xylem vessels, they are dead. They cannot undergo they cannot undergo cell division at all. How about for the bark? Bark, we do not use it as well, because bark is also dead. So actually, what we the cell we need to choose is the living cells. They must be alive. So for example, how about phloem cell? Phloem cell, we will not choose it because they are specialized already. So what the cell we need to choose, it should be unspecialized, and also they are able to undergo cell division as well. So for the chapter 14, okay, if you can really recall the terms meristem, so that's the tissues we need to choose. Okay, the small piece of tissues, it means the meristem from the parent plants. So, and then the tissues, they are grown under the sterile and controlled conditions. Sterile and well controlled. For the well controlled, that means the nutrients, hormones, water, temperature, humidity, something like this, they are all well controlled. But for the sterile, okay, I want to remind you something first. Beware of the meaning of the sterile. 2 meanings. One is germ-free and the other is infertile. But remember, in this case, okay, be sensible. At least for this part, the sterile, in this case, it should mean germ-free. We cannot just mention the condition. The condition is infertile. But the point is that we would like to grow a new plant. Okay, so we put it in a germ-free condition. That means without bacteria, without fungi to harm the plant, right? And then the cell, they undergo mitotic cell division and form a mass of the unspecialized cell. Unspecialized cell, that means there is no cell differentiation occur at all. Okay, no differentiation, only cell division. 1 to 2, 2 to 4, 4 to 8. And then the unspecialized cell, okay, they can develop into small plant, the plantlet. That means the young plant, and then the young plant can be transferred to the greenhouses for the further development. So remember that. Why do we need to supply hormones? Because the hormones, they can stimulate the cells to become specialized, from unspecialized to become specialized, and then they can keep developing and become a mature plant. Okay, so that's the first paragraph. I would like to mention a few key points. And for the second paragraph, it's not that important, but at least you it can explore you more, because this method, tissue culture, is not only limited to the commercial production, but also we can use it to maintain the biodiversity in the ecosystem, because we can regenerate the Asian plants from its fruit. So in this picture, that's the plantlet, the young plant. Once we call it plantlet, at least you can see the root, the stem, and the leaves. So that's one method, the tissue culture. And the other method I would like to mention is that the method of cutting, because in the question we need to deal with the method of cutting and compare it with the tissue culture. So I would like to talk about it now. So for the method of cutting, it has the name cutting propagation. Actually, it's a kind of vegetative propagations, and the example you can also find it in your book. That's the African violet. By cutting, we can also reproduce it. Okay, so actually for this part, we cut a part of the parent plant. Now remember, we cut a part of the parent plant, but not just a small piece of tissues. That's the big difference between cutting propagation and the tissue culture. And after we cut the healthy leaf, put it in water and let the root grow. After the root have developed, so we can put it in the soil to let it develop further and further to become a whole plant again. So actually, in this diagram, in this example, I want to demonstrate is that to show that the vegetative organs are not only the stems, but also the other parts of the plants. But in our book, we only focus on the stems. For example, the stem tubers, the rhizome, the culm, and also the bulb. We only focus on stem, but it doesn't mean that vegetative organs are all stem. They can be root, they can be leaves. But the method, okay, the method is also under these big terms, vegetative propagation. And for the questions, question 1 is the easiest one. Explain why new plants produced by the tissue culture are genetically identical to the parent plants. So the hint is that we call the type of cell division involved in the vegetative propagation is okay. And for the question 2 and question 3 is a little bit difficult. You need to think about it more. So for the question 2, why tissue culture is carried out under sterile conditions. So remember that sterile conditions, it means germ-free. In this case, germ-free, that means to ensure that there are no fungi or bacteria. Okay, so that's the first mark, to ensure that there are no fungi or bacteria. So for the second mark, you tell me, what is the importance of growing the young plants in this sterile condition, no fungi and no bacteria condition? You tell me the reason. And for the question 3, compare with the traditional method like cutting, discuss the advantages of using tissue culture to propagate the plants. So remember, in this question, actually, even you see the word compare, it doesn't mean that you need to list out the difference between cutting and tissue culture, because the focus should be the advantages of using tissue culture to propagate the plants. So you need to talk about is that why do we choose this method? Because this method is better. So what are the advantages? So for the question 3, so the hints is that I want you to recall the specific culture condition in tissue culture, sterile and controlled conditions. So after that, I want you to think about it, the death rate of the plants and also the success rate of the propagation. Why do we use a particular method and rather than another one? Because this method it will not easily make the organism die, or the chance of the success rate is high. So at least that's why we choose one method rather than another one. So 4 marks, I give you the first 2 marks. So the plant is grown under sterile and disease-free condition. So blah 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 blah. You tell me. And the second one is that the conditions for growth are well controlled. The temperature, the humidity, the light intensity, the water, the nutrients, the hormones, something like this. They are all well controlled. So blah 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 blah. So you tell me the second sentence to complete the story for me. So 4 marks, I give you 2 marks already. For the 2 marks, I give you 1 marks already. Okay, very sufficient hints for you to think about it. So in this part, not only to know more the biotechnology, but also you need to think about why do we choose a particular technology to use. Okay, that's something what a science students need to think. In front of us, there are so many methods. So why do we choose method A rather than method B or rather than method C? There must be some reasons we choose. Okay, so think about it and do it first. Okay, bye bye.